start recording. Good. Population ecology. So now we're looking at how to estimate population sizes, the distribution of different species. We already looked, I think, at the red kangaroo in Australia, and it has quite a wide distribution. So we're looking at habitat and range. We're looking at density. Are these very closely packed organisms like wolves, for example, that travel in packs, or are they widely dispersed? Some territorial animals tend to be widely dispersed in a uniform kind of way. And the reason that we're studying population ecology is that if you find yourself at any point being a wildlife manager, a fisheries manager, you'll need to know how to estimate population size. And you'll need to know, is the population declining? Is it increasing? Or is it stable? Or is it on some kind of a cycle? So we look at such things as density and distribution, age structure, and variations in population size. So some populations, for example, have a high number of young individuals and a low number of older individuals. And some populations are the opposite. And you can see that when we look at our population pyramids. We'll look at human populations in different countries and what that looks like. So one question that grad students, and even now this question is ongoing actually, what is causing the declines in sea lion populations in Alaska? So what's interesting is that in the Aleutian Islands and the more southerly coast of Alaska, there are sea lions, stellar sea lions. And some areas the population was declining, and in some areas the population was not. Now my question to you is, let's form some hypotheses here. Whenever you have a question in biology, and this will apply to the study that you do with the animals that you've chosen, either the shore crab or the brine shrimp or the copepods, you'll ask some kind of question about them, like what is their behavioral response to different levels of pH, for example. That's an abiotic variable. So what about sea lions in Alaska? Just throw out some suggestions about why sea lion populations may be declining. What would cause population decline? Excuse me. What kinds of things would cause? Sorry?
A scarce food source?
Food sources. Yes. Good. Food sources can be scarce. They can be changing. And it would depend on whether the animal was able to eat other kinds of prey while the scarce food source rebounds. What is another reason that aquatic populations or marine populations may be declining or fluctuating? What's another reason? Predators. Good, good. What about any abiotic variables that you could think of? It would be an abiotic variable. In the ocean. What's an abiotic variable? pH. Is something changing with respect to acidity in the ocean? Well, we know it is because carbon dioxide, which dissolves in the ocean, does increase acidity. And what else? Could, for example, temperature have anything to do with the situation. We don't know, but that is an hypothesis. So there are many possible hypotheses. And predation, we can break that down perhaps to different predators. So what eats sea lions? What is a predator of sea lions? A very large animal, mammal. It's a large mammal, orcas. So orcas or killer whales, and of course humans. So when I did my study on coho salmon, the question was, what causes salmon declines? And there are many hypotheses. So there was predators, fishing, habitat loss, and abiotic variables in their habitat. So it's interesting. I kind of knew it was fisheries, but the fisheries always try to, not always, I shouldn't say that, that's pretty blanket, but sometimes blame other things for the declines of fish. So I chose habitat myself, habitat loss, looking at coho salmon. So, but how do we know what's normal? How do we know it's normal? Maybe this is a natural decline that happens in a cyclic way. That would require long-term data gathering. Sometimes you can glean data from history, but not very often. So that is a more difficult question to answer. But once we've formed our hypotheses, then we need to test them. In the case of sea lions, it's a matter of gathering information. Gathering information like population trends over time. Have there been noticeable population trends in the past? In biology, and especially in population ecology, we're always looking for trends. And so, sorry, the humans, oh yeah, no, humans aren't predators of sea lions, sorry. Humans are competitors. Thank you for catching that. No, humans are competitors for the same fish, species.
That makes sense. Although I
Yeah.
do remember my dad, he used to be, oh God, I don't know what it's called in English, but he used to be an alpine skier. So he basically used
Okay.
to ski up the mountain and then come down. And
Uh-huh.
I believe that he used some sort of seal skin for better friction
Yeah.
on the snow when he was climbing up.
Yeah. Well, yeah, seals used to be hunted by humans in large quantities. They probably still are. They probably still are. So let's put that back. Let's put it back.
Yeah, I
Human
believe they.
hunters.
Sorry, I believe they use the skin, yeah,
Yeah.
as the bottom for skis to keep traction when you do backcountry skiing. Now they're like synthetic, but
Right,
I know they
right.
used to do that.
Yeah. Yeah. And seals were hunted before for blubber, for meat, and for their fur coats. So, and nowadays, sea lions and seals, I think, as well, they're sometimes hunted to try and keep them away from fish farms, which makes me very irate because, it's not fair. If you put a fish farm out there, you can't expect everyone to stay away from it. And there are other methods to keep away seals and sea lions from fish farms. So those have been tried, like putting in acoustics of orca calls, for example. I think that's met some success. So our information is gathering population trends from the past. So maybe we can try and find out if it's a cyclic event. And we may want to make observations of both areas, the one where the population is declining and the one where it's not, to see if there's any differences in those populations. examine their food source. What do they eat? Sea lions, they eat herring, they eat pollock, and they eat other kinds of fishes too, I'm sure. So those are the kinds of data that you need to collect as a population ecologist. So one thing that the aquarium does look at food sources. So they have sea lions that have been rescued from the ocean. I don't think they have sea lions that they've captured anymore. They've got sea lions that have been rescued at one point or another. And they feed them different types of fish and then examine their poo. And so they can tell which kinds of fish are efficient, have what's it called? Transfer or conversion efficiency. And it turns out that herring have a high conversion efficiency. And that may be because the herring is quite fatty, not really sure, but the pollock do not. And it turns out that, although I don't know if the actual reason has been determined at this point, but the herring are also fished by people. So are the pollock, but the herring are fished quite a lot more. And so the sea lions were switching to pollock. But the weird thing is that, and I'm just recalling some of the data that I heard about from this study, it turns out that the pollock are deeper fish in the ocean. And the sea lions, they only dive to a certain depth and they only stay underwater for a certain time. So pollock for the sea lions is more difficult to get than herring. So that seemed to be part of the problem. Yeah, so that's a bit anecdotal. What is a population? A group of individuals of a single species living in the same general area in which they are likely to breed. If they're geographically dispersed, even if they're the same species, then they're not the same population. And it is the researcher or the observer that decides what that area is. I mean, you may decide you're studying a population in several different areas of the same species. You could consider that your population that you're studying, but it's more common to study a population in the same geographic area. So that's what a population is. Density is the number of individuals per unit area or volume if you're looking at aquatic systems. Dispersion is the pattern of spacing amongst individuals. So how do you measure density? Well, it's impractical to count all members of a population. It usually can't be done. So we use methods such as random sampling, stratified random sampling, and mark recapture. So I'll show you a random sampling example and a stratified random sampling, and then I'll get you to do a mark recapture study at home. It sounds ominous, but it's not. So say we have, I'll give you an example of something relatively simple. Random sampling. So there's a field. It's an extremely large field. Say it's like 100 hectares. And there are different plants in the field. Dispersed. So we're going to do some random sampling. Could we count all of them? No, we can't. So instead, we could make a grid, a geographic grid. It's also easy nowadays because we have, Google Maps and Google Earth and all these great tools. So we've made a grid like that with our geographic information system. And then how are we going to choose what to count? Well, we have to do it completely randomly. And to do that, there are algorithms that you can use in a computer. So you maybe number all of these squares, and then this algorithm will randomly choose numbers for you to go and count. When you're in the field, usually you're just rolling dice, essentially. If you're walking around thinking, am I going to do this grid or that grid? You just roll the dice and you'll choose your grids that way. So there's different ways of randomly choosing them. So we might choose that one, that one, that one, that one. Who knows? It's random. We'll only count those grids. And then we extrapolate those numbers to the whole area, but because we know the number of grids. So this way we're getting individuals per square meter. Often grids are square meters. So we estimate, sorry. We count individuals in randomly selected grids. So if we have, say 10 grids, then we'll have to count the individuals in each grid. So it might be 9, 1, 2, 6. We count those up. Say our final result is 35. And then we divide by 10. And that gives us 3.5. So we are estimating that we have 3.5 individuals per grid, per quadrat. So the quadrat is a square meter. And then we multiply that by the number of quadrats that we have. So in this case, it could be 100. And that will give us total population size times the number of quadrats equals population size. Are you with me so far? Thumbs up if you're with me. Okay, awesome. Let's go to another example. Stratified random sampling. So you might, for example, be counting plants on a mountain. And these plants exist different places. But what is the difference between areas on a mountain? There may even be, say, a creek at the bottom of the mountain. So this is a mountain. These are plants. I should label these things too. This is a large field. These are a grid of quadrats. Sorry, I should be labeling as I go along. These are plants. We're getting estimates of plant size, plant population size. So this is a river. And this is south over here. So this is south. This is north. On this side of the mountain, So what is different about the south and the north sides of this mountain? Which side is getting more sun? Which side is getting more sun? It's the south side. So if we placed a grid on the entire mountain and did random sampling the way we did with the field, it wouldn't work because there are likely to be more of these types of plants or less, depending on whether it's a shade tolerant or not kind of plant on the south side or the north side. So what we do is stratify it. So we'll stratify it. I should have made this more simple. I should have left out the river. But you need to stratify sometimes. If things are close to a river, they're likely to be more abundant than if they're far from the river. So you would stratify it that way. You would stratify it by elevation. And in our case, we're just stratifying by the south and the north side. So we would still do our grids, our random sampling, but we would make sure that there were the same number of samples on either side. So we would randomly sample with equal numbers on both sides of the mountain. And the reason for that, the reason for random sampling is that if you randomly sample the entire mountain, then you could get, just by accident, 2 grids over there and none on the other side. I'm just exaggerating the numbers, of course, and if that were the case, you wouldn't be representing the entire mountain. So that is stratified random sampling. Mark recapture. Okay, good. Mark recapture. This is a different method. So we just looked at plants, but what about animals? Animals move around. They are much harder to count. So let's make a slide here. I'm using the front page, but you could just use a piece of paper that you have. How to count animals. What if they are sessile? What method could we use? If they're sessile animals, could we use the methods we've just used? Yes, we could. I think we could use quadrats, or transects is another method, where say you're in the intertidal, there's all these rocks, and there's lots of organisms. What you could do is get a transect line, you know, and you could count to a meter on both sides. So that would be a transect. let's see, what if they moved around a lot? So motile, lake, shore crabs, or fish, or elephants, or birds. So there are all kinds of creative ways of counting things. Just birds, for example, we do a bird count in Stanley Park, or we used to when we were able to all get together. Hopefully we can do that again soon. I guess somebody's just doing it by themselves nowadays. But what we do is go around with a group of people and we just count what we saw. And we pick a certain area in which we count. There's a Christmas count of birds, I think is across North America, where people are asked to go out on one day and count all the birds that they see of different species. And that's been nationally put together. There may even be an international bird count. I'm not sure. So there's various ways in which to do that. So for fishes, it's usually sample catching by fishing, or the way we did it in streams to catch the small salmon, the fry, is we would electroshock them. And I know that sounds awful, but it's okay. It just stunned them. And then they float to the surface and you grab them with your net. You count how many there are and you release them. There's no other way to count them because they just completely hide under the rocks if anybody's anywhere near them, and you'd never see them. So that was the way to count fish. Elephants, well, how would you count elephants? That's a tough one, but it would depend on whether they were visible or not. And there are elephants that are in forests. They would be difficult to count. But if elephants were on an open plain, you might be able to do it by aerial photography, for example. And even birds, you might be able to count by aerial photography if they fly in flocks. But what about the shore crabs? That's more difficult. With the shore crabs, I think we'll try mark recapture. Which is where you define an area. Say we're just looking at an intertidal area in Spanish Banks. You define an area. You capture, say, 100 individuals. And mark them with something. There are different kinds of completely non-toxic markers that you can use. And with larger animals, you might be looking at tags that you would tag on them. And then you release back into the population, back into their original population. And you are going on the assumption that all these marked individuals will go and distribute themselves randomly and sort of evenly with all the other members of the population. So that is an assumption that you have to make. And then 4, you recapture, say, 100 individuals. So what you've done is you've taken 100 individuals, marked them, put them back in the population. So now within that evenly mixed population, there will be a ratio of marked to unmarked individuals. Marked to unmarked individuals. So, and there will be a ratio of marked individuals to the whole population size. So marked individuals to the whole population size, and that's what you use to estimate the population size. So you've got there's a ratio of marked individuals to population size, which I'm going to indicate as capital N. So when you do your recapture, you should have the same ratio of recapture marked individuals to the total recapture number. So that is the logic behind this type of population size estimate. And so what I'm going to do so that you can understand that better is to give you an exercise. So let me stop there for now. Thank you for watching.